and you put an antisense oligo with modified nucleotides, so that those are stable and those will bind to your boundary, so the splicing will not happen. So you will not degrade the RNA at the mature stage, but you will block the process. Or in those cases, suppose you want to skip some exons, okay, suppose some exons have mutations, okay, and you want to skip those, okay, so you can use modified nucleotide, having like oligonucleotides with modified nucleotide like this, morpholinos, to skip some exons. You know DMD, muscular Duchenne muscular dystrophy. So there is a gene called dystrophin. It's the longest gene. Okay. So in DMD patients, so mutation happens in a specific exon. So people have come up with ideas to skip those exons using modified oligos, so that it binds to the exon and you skip 2 exons and then 2 or 3 exons, and then suppose you are skipping from exon 40 to 45 to 50, so then those few exons are missing, then the other exon is joined now, so you will have a shorter protein, but you will have a functional protein. I don't remember which exon that is. I think 52 or something like that, 50 or 52. So you put modified oligos so that you skip those exons, so the middle part is deleted and you have a shorter protein but a functional one. So those are like therapeutic application where you can use modified nucleotide for the application. And like I said, the gap merge for gene silencing in vivo, you cannot use siRNA, right? Why you cannot use siRNA? You said yes, right? क्यों नहीं कर सकते? siRNA होता है, but you really cannot do it in vivo because that is RNA, and RNA is not that stable. Okay, once you inject, you will have RNAs which can degrade them. It will not be very stable inside the cell. But if you can modify the RNA like 2-oxymethyl or pseudouridine or LNA, locked nucleic acid, you might have heard, right? So those are stable nucleotides, like modified nucleotides basically. So if you inject them into mice or animals, they will stay for weeks, okay, so that they can act, okay. But siRNA will not work for that long. It will work, but for a day, 2 or maybe 3 days, then it will be gone. But gapmer will stay for longer period because of the modified nucleotides, and that is giving them the stability, okay. I think we have all talked about this. Like the transcription happens, okay. After that, RNA is modified, and these are the readers basically. So once the RNA is recognized by the readers, they can be exported out, and then in the cytoplasm, so either some of the modification can go for degradation, some can stabilize them and regulate the translation efficiency, okay. And I think okay, this one is for the disease part. Like if the modification is happening, so till now we are looking at like how these modifications affect the translation stability. So any of the disease, forget about tumor genesis, any of the disease that happen in humans, that is because of changes in gene expression, right? So any time there is a change, alteration in the gene expression, and that leads to anomalies. So the cell doesn't function the way it should function. So that can lead to any disease, right? So I'm just showing one example here, like this is one mRNA, okay, with the modification, the stability is affected, and then you have that this HM box 1 mRNA that regulate the telomerase activity. So when the mRNA is degraded, the telomerase activity is also deregulated. So and tumor happens, one of the like reason for the tumor to happen is because of shortening of the telomere. Okay. So telomere gives the stability. You know how telomere is synthesized? It's there on the slide. Huh? Which enzyme synthesizes the telomere? Huh? Reverse transcriptase. So that is a telomerase. Okay. So that is a protein, DNA or RNA? Telomerase. Huh? DNA. RNA. RNA protein complex. So this is basically RNA act as a template, okay, so that the telomerase can synthesize the telomere and make it longer. But in cancer, this doesn't happen that efficiently, and then you have genomic instability that leads to other chromosomal misarrangement or translocation that is giving rise to tumor genesis. Okay, and the cap modification, you all said which modification is the cap? G7, M7G, right? Huh? Okay. Then the other modification, this is the canonical modification, like you said, that's there in the book, right? The other modification type, they include 2-oxy-methyl modification also that happens. And here you see NAD, FAD, ADP. So these are like minor modifications, but these are not canonical cap. So they are also used as cap in the mRNA. Anything you want to say? No. Okay. And then the non-coding RNA. The coding RNA, we have seen like that any of the modification on the mRNA that affects mRNA stability or translation. And if the modification happens on non-coding RNA, what will happen? It is not getting translated, right? Yes or no? Then what will happen? Why? mRNA formation. No, we are talking about link RNA. So link RNA doesn't code for protein, but it has other functions, right? I said link RNAs, they regulate RNA binding protein activity, microRNA activity. They can interact with the mRNA. So if you change the link RNA modification, so the stability of those link RNA will change, correct? So with that, it will affect the gene expression indirectly, not directly. But in mRNA modification, it is direct. Your tRNA modification, rRNA modification, they affect directly the translation system. But link RNA modification, they regulate gene expression, but indirectly, not exactly directly for the gene regulation. And this is the M6M machinery. We have talked about writer, eraser, and reader, right? I will not go through this. And maybe I will share the paper with you. I don't think how many of you can read this slide. Nothing you can read, right? So the number of modification we talked about is, one is adenosine, one is cytosine, M6A or M5C, and then uracil, that is modified to pseudouridine, and what else? 2-oxymethyl RNA, and then which one? A2I, adenosine to inosine. Okay, so all these modifications, and then there are many more here. Okay, and M1A, 2-oxymethyl, and this is M7G, and this one HM3C, this is 2-oxymethyl again. So these enzymes you see here in the blue, okay, so those are the modifiers, okay, and then this are the readers, and then erasers. Where is the eraser? कहाँ है मुझे टॉप? हाँ, ये जो राइट साइड में है, those are the erasers basically. So there are many modifications, not many readers or writers, few of them, but they are involved in modifying many RNAs. Okay, and how do you detect them? Any method? We have some time, right? Okay. If something is modified, how will you check them? Sequencing. Okay. How will you sequence? Suppose DNA methylation. Chalo. How will you read that methylation? Sanger sequencing. Okay. Sanger sequencing will read the modification. I don't think so. No. Ataxic. Ataxic will not read modification. Next generation is like basically high throughput sequencing. Okay. So how will you read that? What is the difference between Sanger sequencing and next generation sequencing? Anyone knows how sequencing works? Okay, how?
We use specific probes, pre-designed probes, and in next gen sequencing we have a module in which different color fluorescence is released for different sequences, and then that is read by the computer, and then we get the positive right.
बैठ के भी बोल सकते हो, but Sanger sequencing also does the same thing. Sanger sequencing also have the fluorophores. Without that, how the machine will read it? It is not because of the probe, because of the fluorophores only, the modified nucleotides with color. Okay, so that is also same way, like chain termination reaction. So when the modified nucleotide is incorporated, the chain will terminate there. Okay, so you will have different length of nucleotide with the modified color, right? Modified nucleotide with the color. So it can be A, T, G, C, depending on wherever the end the polymerase is incorporating the modified nucleotide. It is a random process. So most likely you will get fragments at each location. Okay, so that is how Sanger sequencing work, because you know the sequence and you have the template. So you can align कि कौन से position में A आ रहा है, T आ रहा है. So the machine will give you the sequence. But that is done for only one target. That is the major difference. You can have only one single target and do Sanger sequencing. But in the next generation sequencing, what happens is you have millions of sequencing done at the same time. Okay. Have you seen a sequencing machine? कौन सा वाला? Sanger sequence? हाँ? Illumina sequence. कौन सा machine? Illumina is the company name.
Know that particular name of that model.
Okay. Next generation sequencing. Okay. So they have NextSeq, NovaSeq. So NextSeq is the smaller version. NovaSeq is the higher model. So in these sequencing machine, suppose you are using NextSeq, you can have 400 million reads at a time. In NovaSeq, it can go to 800 million reads. Okay, so in Sanger sequencing, you are just reading at one sequence at a time, but in the next generation sequencing, millions of reads are sequenced at the same time. Okay, and how the machine detects in Sanger sequencing? Any idea? हाँ, कैसे रीड कैसे करेगा कि इस पोजीशन?
पर्टिकुलर साइट लाइक वी यूज फ्लोरोसेंट टीटीपी, वो हर एक के पास रुकेगा, हर एक फ्रैगमेंट, शॉर्ट फ्रैगमेंट, लॉन्ग फ्रैगमेंट.
हाँ, संगर सीक्वेंस नहीं, कॉम्प्लीमेंट्री सीक्वेंस नहीं होता है. कॉम्प्लीमेंट्री सीक्वेंस इज रिक्वायर्ड फॉर सिंथेसाइजिंग दैट सेगमेंट, लाइक यू पुट ए प्राइमर then go on synthesizing. So that is the process. So you have a primer, then the primer is getting extended with regular DNTP or the modified DNTP. So wherever the modified DNTP is incorporated, that has a collar and it terminates. So that is a chain termination. So you get the fluorescence there. So you have hundreds of reads at a given time, right? So you run them on a capillary. Depending on the length of the DNA sequence, it pass through. Like your HPLC. जेल चलाते हो ना? किसी ने जेल चलाया अगरोज जेल? ओके, तो अगरोज जेल में कैसे सेपरेट होता है डीएनए? साइज के बेसिस पे. सेम वे in Sanger sequencing also they are separated. So the smaller one will go first, then followed by the other one. So length wise the machine is reading कि कौन से न्यूक्लियोटाइड कब पास कर रहा है उधर से, ठीक है. So then the machine will tell you कि चलो ये A है, T, G, C. So based on that, based on the like fragment size, you get the reading. And because of that sequence, whatever is coming, you align with your target DNA and then you get the sequences. So and then confirm कि okay, the sequence is there or it is modified, mutated from the genomic sequence or not. But when it is for the next generation sequencing, you have millions of reads. Okay, how will you do that? Like find out कि कौन सा कहाँ है, किसके साथ align करोगे? Reference genome के साथ. Okay, so genome or transcriptome, whatever is there, you align and then look at, suppose you are looking at gene expression, you are putting those 400 500 million reads, aligning with the genome and seeing at which position how many reads are aligning. Based on that, you will see suppose this gene has 50 reads. Like suppose you imagine this is your genome and these are specific genes. Okay, and in this row you have like 10 people. So 10 reads are stacking. Here 20, here 15. On the other side you have 5. So based on the number of reads it is stacking on each position, you can say how many genes are actually like how much one gene is getting expressed or not. And if you have two classrooms, okay, and you separate them by saying this is gene A, gene B, gene C, gene D. In the other classroom, you have a difference in the number in each of the pattern. So based on that, suppose this row instead of 15, on the other class you have 10. So you can say, okay, there is a down regulation of that gene. So depending on how many reads are coming for each gene, you can say which genes are expressed more or less. Suppose any disease or tissue, suppose you take a skin cell, okay, you do the sequencing, and you take suppose muscle, the sequencing will be completely different because the genes they express are very different from each other. So the reads you get are very different. So based on those pattern of read numbers per gene, you can see which gene is expressed how much. So next generation sequencing is mostly for the expression checking and for modification also. But for modification, you cannot just go and put the RNA and say, okay, what are the modification? You can read them. You need to do some chemical treatment or you do enrichment. Okay. So like I said, M6A modification. So if I say, okay, this gene has M6A modification, you need to pull down with antibody, M6A antibody, and whatever RNA comes with that pull down, you go for sequencing. So that way you will find which genes are having M6A modification. So for the other modifications, we don't have antibodies till date. But M6A is the one which you have a very specific antibody, and lot of people have used to find which genes are M6A methylated, because most of them are methylated. To find the position, there are chemical methods where you can modify the nucleotide and then go for sequencing to find that out. Okay. So either antibody based, like I said, then mass spectrometry. You know this technique? Huh? How that works? Based on the mass and charge ratio, the fragments move and the machine reads this is the mass of the fragment, and based on that you see whether something is modified or not based on that. And then chemical treatment, like the modified nucleotides are treated so that they are modified again and they incorporate mutations. When you do the cDNA synthesis, so if they are modified, they will not have exactly the complementary sequence when you are making the reverse transcription, and then that gives rise to mutation. Then you do the next generation sequencing, align with the genome and see where you are getting those modified, like mutated sequences. From that you can say, oh, this position is actually mutated. And if you are comparing two disease condition, in one you can say, oh, in one condition you don't have this mutation, in the other condition this specific gene or specific position is mutated. Okay. So like I said, the dot blood, this is antibody based. Suppose you want to see M6A methylation in two different samples. So you want to see in cancer versus non-cancer. If you say whether M6A methylation is more or less, you can have two RNA population from equal amount, of course, and then probe it with the antibody. Then go for Western. Have you done Western? Huh? Very few. Okay. So basically you put the antibody, then put another secondary antibody with a HRP. Okay. Not fluorophore, HRP. Then you put with luminescence reagent, you get the light. So if you want to see in two different conditions how the modifications in the RNA is different, probe with antibody. And in one case, suppose you say very mild light, and in the other one you have a glowing light, so you have darker band dot. So you can say, okay, in this sample the RNA are heavily modified, right? And the mass spectrometry, as I said, in different fragments you can see whether some RNA is modified or not. And I will not go into the details. Okay, as I have explained, like either you can modify with chemicals or with enrich with antibody, then go for sequencing. I will not go into this. So yes, in one way you can do is you modify the nucleotide so that either your reverse transcriptase incorporate a mutation or reverse transcription stops there. Okay, so the RNA is modified now, so the reverse transcriptase cannot move forward. So wherever there is a mutation, the reverse transcription will stop, and when you go for sequencing, you will see the RNA stops, the fragment stops there itself. Okay. And you can do PCR, and this is another way, like if something is modified, you put two fragments, ligate them, and then do qPCR. So with or without modification, the amplification will be different based on whether they are ligated or not, like the probe you were mentioning. So that way you can do it. And then this is qPCR again, this is mass spec. Yeah. So yeah, so suppose here you have A to I modification. When the reverse transcription happens, I is converted to C. Ideally, it should have been A converted to T, right? But now, because of this modify have a C, and then when you do the sequencing, instead of A, you will see what? G, because in the cDNA you have C. So when you actually match with your sequence, it will be G. So instead of A, you are seeing a G in the sequencing. So that tells you there is a mutation at that specific position. Okay, this is one way. And like I said, either you can stop by modifying the nucleotide so that the reverse transcriptase doesn't cross that modification, stop there itself, and then you see from the fragments, this is where the modification is happening. This is again chemical modifications. I will share the papers, maybe you can read to go through this. This is another way, same way, like you modify and then in the sequencing you see the mismatches and then you read it. And the other way to read, we have 5 minutes. Okay, so the other way to do sequencing is nanopore. Have you heard of that? Nanopore sequencing. So why the name nanopore? The size of the pore is in nanometer scale, so it's a nanopore, and you sequence when the RNA or DNA pass through the pore. Yeah, so when the nucleotide pass through the pore, they generate some electricity. So based on the changes on the electricity, you read them. Okay. This is the enzyme. There is a motor actually attached to the one end of the RNA or the DNA when you make the library. Then the motor makes the RNA pass through the nanopore, and the like here the program detects the signal. Based on the signal, it will say, okay, it is A, T, G, C. Or if there is any variation in the sequence, like the electricity, it can say, okay, there is some kind of modification is there. Like here. So if there is a change in the electricity, you will see there is a modification here. Okay. And as I said, like if you make the library, so library one of the strand have the motor addition, the adapter has the motor addition. And if you go for okay, so this is cDNA. For the RNA, you make the cDNA at the adapters, ligate, and then this whole thing goes for the sequencing. But only strand with the motor protein, they go into the pore for the sequencing, not the other strand. Okay. Okay. So yeah, and when the RNA pass through this pore, so the electric signal is read by the machine, and there are programs which can tell you which modification is there. So for over the last like 8-9 years, several softwares or the algorithms have come up with modified like protocols to read what kind of modification are there in the RNA. So this is a kind of direct sequencing where you can see the modification. And the challenges is like we talked about around 300 modifications, but hardly for any sequencing for that antibody based chemical modification or for this nanopore, you can hardly read maybe 10 modifications. Others you cannot do it in high throughput. So that is the biggest challenge. Like they are there, but you cannot do it for all kind of modifications, and their role is not known for every modification because these are dynamic. Once you sequence, you get the modification with changes in the environment or physiological or changes in the expression of the eraser or the writer. Those modification will change, and these are very dynamic. Because of that, you cannot like do sequencing at every time point to see what kind of modification are happening. Okay, and these modifications are costly to the cell because regular nucleotide you are just incorporating by transcription, but when you are modifying something that needs energy, and because of the modification the RNA stability or the changing, right? So these changes, the changes in the gene expression pattern, structural changes, and the also the processing. Like if the modifications are happening in the pre-mRNA or the snRNA, so those can affect the splicing also. Okay, so you can affect splicing, stability, and translation efficiency with the modifications. And in future there should be more methods actually to find out what kind of modification is happening and in which condition which modifications are more prevalent and what are their functions in the disease perspective. So those things are like the future where we will be knowing more about the RNA modification. As I said, like there are 200, 300 modifications, but we hardly know like 2-oxymethyl, M6A, pseudouridine, like only few of them are very well studied. The writer, reader, and erasers are known, but for the other ones, it needs lot more research to find how they are happening and what are their functions in the cell. And this is my group. So this is in ILS, and my lab is actually funded by intramurally by ILS. This is Wellcome Trust. They funded me for studying RNA-RNA interactions